let's ask some questions about Carl Linnaeus. First of all, who was he? Where was he from? And what century did he live in? Where was he from? Sweden. What century did he live in? The 18th century, meaning what? The 1700s. Did he come before Charles Darwin or after Charles Darwin? Before. What did he attempt to do as one of the main things in his life? Yes.
Classify all the organisms into a simple coherent system.
In a simple coherent system to classify all organisms. And that was called what? What was that system called? Called it by a name. I don't think no. Let me go back. Can we get my marker here? Right. It's not the binomial system. No, he called it something else. The lady who was talking about, there was one woman in the video. What was she speaking about? She referred to the whole classification system as an 18th century version of what? Excel spreadsheet. And that whole book that she had was called by a name.
It's like something
System of nature. In Latin, what? Right, Systema Naturae is what it was called. And a natural system of classification. What exactly is a natural system of classification?
It's when you classify things based on like relationship to a common ancestor rather than traits.
Right, so you say classifying things in relationship to a common ancestor. But do you think when Carl Linnaeus went out classifying mainly what? What evidence did we have in the video? His main thing that he was focused on was what? Plants. So he went out classifying plants. Did he have evolution in his mind, that I'm classifying these things to show how they evolved? So his system is natural, but it's not with evolution in his mind, because evolutionary ideas, as far as we know, didn't exist in his time. In fact, he was one of the famous biologists before the time of Charles Darwin. When Darwin and Wallace had to go present their research to biologists in their time, they actually had to go to a place called the Linnean Society. So Carl Linnaeus was like one of the famous biologists for them. And the society that they had to go present to was called the Linnean Society. So Linnaeus was famous for his work with Systema Naturae. And it was a natural system based on him going out and looking at plants and looking at what the plants look like, saying that they all have certain features in common. Let's put them under this group. So if they all have flowers, let's put them under a group called the flowering plants. And if they all have if they all have cones, let's put them under another group that we might call conifers. These have specific names, which we will discuss later. Right. Flowering plants, conifers, a bunch of other things. He's deciding what goes into each group based on what? Based on certain traits that he can observe on the outside. Now that we have some understanding about evolution, is it going to be a problem if we say Linnaeus put all these things in groups based on having common traits? Is that necessarily now going to be contrary to evolution, against evolution, now that we have all the evidence for evolution? Or is it likely to be, for the most part, a good match with evolution? What do you think? Mostly a good match, but there will be conflicts. That is a very good answer, but for it to be excellent, we need what? We need examples of evidence. So tell me about some example that you can think of that was classified by Linnaeus some group that you might know about. They've been classified under one of his groups that he identified in his hierarchy, which I will mention in a minute, but I know that all of you have some knowledge of this before. So, could we think of any group that was put together in the Linnean system, that now that we know about evolution, all those members still belong together? There are many examples, so let me see if anyone can tell me an example. All right.
All but like most of the trees together, probably all together as trees.
Trees. Flowering plants is mentioned here, but let's take our group, the group that we belong to. Could we think of one big group that includes all of the monkey-like animals? What do we call that group? Primates. Primates were a group that was decided upon with the Linnean system, Systema Naturae. It was decided that everything that looked monkey-like would go under the order Primates. Now that we've got fossils collected, we've got genetic evidence collected, we've got all kinds of supporting evidence, don't we still believe that primates all share a common ancestor in the tree of life? Right. So for the most part, this Systema Naturae that Linnaeus came up with, it's still very much in line with evolution, which is what biologists have been thinking about doing with classification since his time, and after the time of Darwin, going into the 20th century. They were then focused on phylogenetics, systematics. Which is about classifying all life in a way that would show their evolutionary history. The Linnean system wasn't really built for that. It was built on a natural system where we look at what things look like and say, they all have this in common, so let's put them under this group. They all have that in common, let's put them under that group. The more things that a group has in common, the more closely it would be related in what we call his hierarchy of taxa, which includes what? Kingdom, phylum, class, order, family, genus, and species. And we'll discuss that since his time to now, we've also added something called, recently, the domain. To help us remember it, people put some simple words to go with each one. Has anyone ever heard of anything? Maybe you might have heard of, well, you never heard of it. Dear King Philip came over for good spaghetti is the one that's most widely known. Right. And then I've suggested also that, well, if you don't want to be culturally biased, you can change it from spaghetti to sushi or to whatever you want it to be. The King Philip is coming over for, right? Or if you want it to be culturally neutral, then you can say he came over for good seafood. So you don't have to say spaghetti or sushi or anything, right? So that's international mindedness, which is what IB students are supposed to have, right? So, yes. So, dear King Philip came over for good spaghetti, good sushi, good seafood, whatever you want that to be.
I just got that.
Yes. Right. Oh, you just got it with. Right.
Wow.
Right. Okay. So this is called a hierarchy of taxa. Why is it called a hierarchy? What does that mean?
Comes in order.
Right, it goes from a big group that includes a lot of different things, then into another group where you keep narrowing things down until you get all the way down into the group that you want. Right? So a biologist that finds a specimen nowadays, if he finds a fish, for example, and he's a biologist, he would already know where that goes on this hierarchy. He doesn't have to go say, oh, is this a plant or is this an animal? Where does this belong? He already knows it's a fish, so he's very likely to go straight down to a level like the class and know what class that fish belongs in. And then he may not, depending on how specialized he is in that area, he may not know if it's what groups it goes into after that. But we've already built on a skill that could help him if he's out there trying to identify this fish that he's found. What tool could he use? An identification key or a dichotomous key is what he would then use. That doesn't have to begin from the level of the top of this hierarchy. You begin from the level of your expertise. And usually if you're studying snails, you already know that snails belong to a certain group, and you may go, depending upon how specialized you are, you may go straight to the genus or you may go straight to the family. Usually you'll go to the family. And then the dichotomous key will take you to the genus, and if you really have a good key, it would take you to the species that you're looking at. All right? So this is the hierarchy of taxa. This is what Linnaeus came up with. But then one of the speakers on the video says that Linnaeus's primary legacy was something else. His main legacy, the one that's been agreed upon by biologists, that they will continue to use it throughout the 21st century. What is that? What does that take away from the time of Linnaeus that we still have today? What is it called? No one knows. Yes, Eric, do you know it?
Yeah.
But we all wrote up our lab. What was our lab about? But I hope that we all called it by what? Yes. Why did we call it this? Why did we call it Vigna radiata or Vigna radiata? Actually, I'm not sure which is the one that you should say. Right? Why did we call it that? I heard some answers. Yes.
Different dialects. And we have so many dialects in a single language, and we have so many languages in the world that it gets really confusing when we try to talk about one thing that's different languages.
Right. So communication then. One of the main reasons that we have this is for ease of communication. Right? This naming system that Linnaeus came up with, called what? What was it called though? The binomial system. Why was it called the binomial system? It's made up of two parts. What is this part called? The genus. And what's the second part of the name called? Genus and the species. Why did I underline it? The significance of underlining it always, especially when I write on the board or I write by hand on a piece of paper. Okay.
Make it like separate from names that are used.
To make it stand out. That's protocol, that's procedure. But if you type it on your lab report, how do you write it? Or did you write it? You write it in italics. All of these are things that have been agreed upon by biologists through a series of congresses. Right? These meetings that biologists have, international meetings to come upon common agreements for ease of communication. So right now we are in the process of looking for lichens around the world with a group that begins here in our class, but we are trying to extend that group to schools in other places and students in other places. And one group's joined us and they've collected data for us. In case you don't know, you should check out and see a school in Illinois, right? And
Is that the teacher posted?
yes, so there's a teacher posting her icons is an eagle also, right? So she's posted some pictures there for us, and she's following the same procedure that we're following, so that when we're done, we can decide when we look on tree trunks of tall trees all over the world, do we have a correlation between air quality index and the presence of lichens? That's what our refined research question will be, because we've all been looking for that. And then Mr. Horton. How many of you had Mr. Horton?
Yeah.
Right. Mr. Horton has decided to help us. And I'm glad that he replied to me this morning, and he only criticized me about one thing on the procedure. I was like kind of bracing for him to criticize me about lots of things, because he's a very experienced field researcher, a photographer now, and he's traveled all over. He's seen lots of things. So he told me this morning that I should think about like what side of the tree we're taking the picture from, whether it's north or south. He said that could make a big difference. So I said, well, let's add that on. as something we should do,
Oh, whoops.
right?
So are we
Adding something onto the procedure is not such a bad thing. But it's when you modify it, that makes it hard now. Because you've had some people that collected it that way. But you're adding on one more thing. You're asking, because we were just saying, well, you look at the tree trunk all around. Does it have lichens? So that's nothing. If you're now saying, well, tell me what direction you're facing, and if you only got it, because it's possible that you could get it in all directions. Or it's possible that you might just get it in one direction. In which case, if you're only getting it from one point, that could be something else for us to have an extension, another research question. After that, why are they found on certain sides of trees? So protocol is important in science for communication, especially if it's a lab procedure. And of course here with communicating about species, because you found this snail and you want to know if you're the first one to find it. Or you want to find out more about this snail, where it's come from, if it's an invasive species, one that doesn't belong here. And you want to go to the literature and start looking up things about it. And suppose a scientist in Saudi Arabia has written about this. The script that he's writing in to begin with might not be even Roman script, but he will still have to communicate Vigna radiata like this somewhere in his paper, because that's been standardized. Right? We've had some Korean students in here last period. We still have some here, but I don't know if you guys know. They told me that if you're writing this paper in Korean, you still have to have the biological name written like this.
Yeah, they always have it in the
Right? So you check it out. You guys have had that experience. I haven't. Right? Seeing the paper in another language with a biological name still written in the Roman script in this Latinized way with the genus and the species. That's for standardized communication. Right? That's the reason. And it makes it so much easier. It's already so hard. You're looking to find out what species you have with keys and all kinds of things. It will be totally complicated if people who speak a different dialect within the same country, like you said there, even within a country like India, where people have different scripts and different languages within the one country, then that would be very difficult to communicate that way. But this is standardized. Right? So the protocol is standardized. And that was Linnaeus' main legacy. And now through a series of congresses, we've decided to keep this binomial system. There's a good reason. It's not just like to honor Linnaeus, which is how we could look at it. He was a great biologist. But there's a bigger reason why we should still keep this way of naming it, even though we might change how we do the hierarchy of classification, which I will talk about in a bit, why this is going to change, and it's already changing. But this should stay for a good reason. Besides keeping Linnaeus' legacy, what's a good reason for keeping it well into the future of biology? Anyone tell me?
Well, it's depending what they're going to change it to, if they were going to change it.
Well, what they're going to change it to, that's a good point, yes. But
I think it's like very organized. Like just the first part. Like,
well, not this. Well, this one's in change right now, but the binomial system.
It's already like well documented. So if you change it suddenly, then like everything before would be harder to make.
Right. I think there's some cities around the world where the name's been changed. That's pretty straightforward, right? So that, like, let's say a city in India, like that's called Mumbai, used to be called Bombay. So that's okay. People know that. It's kind of sounds similar. It's different though when you're looking up a research paper now. You're working on mung beans and it's now called something else. Now people have done 50 research papers some time ago where they called it something different.
Yeah, you can't find it.
But you don't know, you have to now, that's one additional piece of research that you have to do. What was its former name? And then, so you've, in one sweep with its biological name, you can find all of the research that's on it. Now you'll have to do two, right? And that's just an added piece of confusion there. And it's the same thing if you're a social science student and you don't know that a city's name's changed. Then when you search online, you may be missing lots of things if you don't know that information. So that's not necessarily a good thing for that reason, but of course that's a different area altogether, and there are social reasons why people change the name of a city. And biologists don't have those kinds of attachments. We've already agreed that we're staying with this system of naming. Even though people have different ways of writing, we've had this common agreement. So since the time of Linnaeus, biologists felt the need to to place things into some hierarchy that would show evolution. This is called phylogenetics. Right? And the Linnaean system was built before this. So since that time, they've been looking at organizing the tree of life to show all of the evolutionary history. And in his time, he only identified 2 kingdoms because, like it says on the video, he was an empiricist. He only went by what he saw. And going by only what you can see in the 1700s leaves you with only a couple things that you can see in terms of the diversity of life. So he put life into how many kingdoms do you think? 2. What are the 2?
Animals and plants.
Plants and animals.
At his time, I believe that we didn't even know why. I'm not sure when. When did that experiment happen to figure out why meat rots? You guys know about that experiment?
Well, they put a cover over the meat.
Right, and it was rotting?
Spontaneous.
Right. So I'm not sure. I have to look up and find out when did that experiment happen. But people didn't really know about microorganisms at all. Why wouldn't they know about microorganisms at all?
Was that before?
You can't see them.
That might have been the 1400s. That might have been before. But why didn't people know about microorganisms?
You can't see them.
It's not visible. They can't see it. They can't see microorganisms at all. Right? So plants and animals were all that we had there. And then the light microscope came around, and then the electron microscope. And classification began to change. Things that we could see that were being placed under these groups, we began to question them as well because of microscopic evidence. So in time, by the midway towards the end of the 20th century, biologists had recognized some more kingdoms. Kingdom fungi was recognized.
Sorry, 1600s.
The 1600s. So it wasn't too long before the time of Linnaeus. The kingdom Protista was recognized. And then the kingdom Monera. So you're looking under the microscope and you're seeing bacteria. And you're studying these bacteria. And you believe that they all have something in common. There's some special type of cell structure that bacteria has that's different from our cells and plant cells and the cells of a fungus. And I know you guys did cells in 10th grade already. So yes, Kinbyu, what is it? Prokaryotic. What defines a prokaryotic cell? They're single-celled, yes. Bacteria can live in colonies sometimes, small colonies. So it's something else that defines it, not just single-celled. They tend to be single-celled a lot. They have asexual reproduction, but in terms of the structure of the prokaryotic cell, who's saying no nucleus? Yes, Eric, that's correct. So that's the main thing. And we did not begin our study from topic 1 because you guys come with some knowledge of biology already, so I don't see that we had to begin from topic 1. And there you go. We already know what a prokaryotic cell is. It's a cell that doesn't have a clearly defined nucleus, among other differences, which we will discuss later. And then the eukaryotic cell has a nucleus. So that was a powerful piece of evidence that allowed biologists at their congresses to decide, well, look, let's put Monera for all the bacteria. They're all prokaryotic cells. That was a very good reason. Then they're looking at the fungi. Fungi are a group that includes a main type of what? What type of decomposer that we study about so far? We've studied about saprotrophs. They have a lot of saprotrophs in there. Right? They feed in a different kind of way. It includes what?
Leaking out there.
What is the most common type of saprotroph that we have studied about in terms of everyday? Mushrooms. Do you know the answer to the question, why is it that a person who claims to be a vegan does not only feed on the plant kingdom? Because they're very likely going to be eating mushrooms. Also, they're going to be having bread, which is made using what? Yeast, which is? No, not a bacteria. A fungus. And then people have mentioned to me today as well that if you're going to chomp on your piece of broccoli, it's teeming with lots of bacteria.
Wait, so they can't eat anything?
Wait.
No, well, it's no vegans out there saying that I only eat plant material, but it's a technical one that you can throw out there at them. Do you think you're only eating plant material? No, you're not. You're eating fungi and you're eating bacteria. Right?
The thing on top of the top part.
No, the thing on top of broccoli, it's all plant material.
No, but you said there's something that's teaming
Yeah, it's probably
Teeming with bacteria means the broccoli itself.
Oh, then like, oh, I see what you're saying.
Right? The broccoli itself, you take a swab and you rub it onto the broccoli and you put it on your plate, it will grow bacteria for you. Right? There's bacteria living on your clothing,
But there's bacteria on it.
on your skin, it's everywhere.
Like if you drink water,
And most of it is good. Last year when we had the Nobel laureate come here, he spoke to us about bacteria. Right? And I also give an assignment in class sometimes, a day without bacteria or a month without bacteria. What would happen in the world if we could banish all bacteria? Would it be good or bad? And it's going to be a lot more bad than good. So lots of bacteria are kind of neutral. They don't do us anything. A few of them are pathogenic. And of course, we need a lot of them. For things like what? What's the first that would come to mind? Well, digestion, yes. What about decomposition? Right? They're important decomposers, and then they play a part in the carbon cycle and the nitrogen cycle because of this role as decomposers. So there were 5 kingdoms for some time, and then there was a biologist called Carl Linnaeus, who, I think I spelled his name correct, the spelling is correct there. He came up with the idea that the kingdom Monera, with the basis of it being everything is what? Everything in Monera is supposed to be what? What is it, Danny? Prokaryotic. That's a good, strong biological reason to say all these things belong in the same kingdom. But Carl Linnaeus was studying the genetic makeup of these bacteria. He was studying their RNA sequences. When he studied their RNA sequences, he found out that the RNA of one kind of bacteria was completely different from another kind of bacteria. So then he realized that there was one type of bacteria called the archaea that was more closely related to the eukarya than they were to the other types of bacteria. So this was based on RNA evidence. So based on this RNA evidence, Carl Linnaeus decided to tell us, well, he told us that bacteria belong in their own group. Archaea are actually more closely related to eukarya than they are to bacteria. That's what this phylogenetic diagram is showing us here. That there is a common ancestor where life on Earth begins. Then archaea evolves, and the fossils going in one direction, bacteria in another direction, and then from that, all the eukarya that we have here, which now includes 4 kingdoms: the plant kingdom, the animal kingdom, right? The protist kingdom, and the kingdom fungi. They're all eukaryotic. They belong up here. All the eukaryotes. So we've got these 3, and this is what we call the 3 domains. So we'll be studying about the 3 domains, and your assignment today is to see what time we have there. Here till 1 o'clock. So this will be the assignment like over the next 2 classes, because this is the application that we are here to focus on. All of that discussion that I just had there is taking in a lot of the understandings, which I have not listed here. Right? But this is the application. This is the evidence that we want to have of our learning. Classification of one plant and one animal from the domain to the species level. So we are going to go into the mesocosm maybe. If you don't want to open up your self-sustaining system, if you don't want to open it up yet, you can leave it like this, and then you can take some plant from my open system if you want. But you're going to take like a moss or some plant that you don't know what it is and try to identify it. As you try to identify it, you are going to create a document putting the image first of the actual thing that you're touching, and then you're going to list its domain, kingdom, phylum, class, order, family, genus, and species. Then you would name the domain, name the kingdom, name the phylum, and then give some reasons as to why you're putting it under each one. So in doing so, you will get some learning, and it will also count for something to be in our practical scheme of work, because we're actually doing it with a real specimen. Okay? Will you be able to get down to the species level? Let's say you get down to the family level, and you're not sure where to look on Google. What would you do for the specimen that you have? The genus, then, let's say. Yes. You search for a key, because we've worked with a key last time and we found the identity of the algae. That's really where the key would become useful. Using keys to classify at the kingdom level is just going to give us the concept of a key. No one in biology needs to classify, needs a key to find out if something is a plant. Although we might have our doubts about if algae are plants. Algae are not plants, actually. They're put now under the kingdom Protista, right, for certain reasons. So the idea that the application is one plant and one animal, that restricts us to one domain only. Does that make sense? We're restricted by this, the way that this is stated, to one domain. What domain is that? It's eukarya, because plants and animals belong to the same domain. Plants and animals are so closely related. When we look at the diversity of life, there's so much of that other life out there that we are actually close relatives with plants. It's hard for people to see that kind of thing. We see the world through our eyes, hearing what we hear, seeing what we see, judging everything that's big like us as being important in the world, usually, and judging everything that's long or short based on our human lifetime. But the idea that an intelligent being could be sitting in this room right now, and it could be microscopic or very tiny, and it could be sitting on my glasses right here, right now, collecting data, that's going to bring smiles. But I don't know that that's impossible. That's just beyond our current understanding. The idea that an intelligent creature could be in this room collecting data, and it could not be reflecting visible light. So it's in this room, it just does not reflect visible light. That's a possibility. The idea that it could be screaming right now at a frequency that we can't hear. It's also a possibility. All right? So people do have these kinds of ideas, and they go set up infrared cameras and UV cameras, and they have things that can record all kinds of frequencies because they're searching for the unknown and trying to collect data. I don't know that they've found anything yet. They haven't even found Bigfoot or the chupacabra or any of those kinds of creatures yet. But we tend to think about things like just from our perspective. And the reason I bring that up is because there are two other domains of life which are very, very diverse domains of life. It's just that we can't see them. The domains archaea and the domain bacteria. And there's a whole load of stuff to even discover in there. All of the mammals that live in the world haven't been discovered yet. People are still discovering mammals and birds. So we can't even begin to think of the diversity of bacteria, archaea and bacteria, true bacteria. How much diversity there is to discover there. So if you want to discover a new organism, there are lots of them still to be discovered. Right? So all of this will be classified for your plant example. And then for the animal example, we'll try to go in reverse for the hierarchy. Although you can choose whatever animal you want, we are going to choose the mammal that has the highest population in this room right now. Which is what? People? Are you sure it's people? Are you sure it's humans? Nope. It is. It is musculus. Yes, that's mice. There are 30 of them living in that box. And how many of us we have in this room here? There are more than us. Their population is more than ours. But you can also choose the ghost shrimp if you want, which we have in the, if you want to choose the ghost shrimp. Or you can choose Daphnia, which is living in our aquarium. You have to choose an animal that's living, that you can bring as a living specimen, take a picture of it. If you really want to go with Daphnia, I can give you a microscope camera and you can take a picture, but that's going to take up so much time beyond the, and it's not necessary right now. So go with the mouse, mus musculus, So you already plug in as genus and species, and then you go up the hierarchy for the mouse. Again with a picture. I have cameras if you want, and again, a good idea to hold a mouse if you've never held a mouse before, right? We keep mice in here, but we're not ever cruel to animals because IB does not want us to break the animal policy, right? And you know the whole world is listening to me here. So.
Fine.
Right.
So like no torture. What about like effective?
A student who wants to work with animals doesn't mean you can't work, but you must reflect on how are you going to be humane, and the idea is to treat the animal better than it is in the wild. So if you want to see how putting a mouse in a luxury home how it would behave, you can do that. Like we had a student before who wanted to give the mice a very healthy diet to see how that would affect their weight and their respiration, and that was approved because we were taking the mice out of this classroom and putting them into his house, where he was even giving them a lot better treatment than here. Right? And I knew that because his mom had all these kinds of air fresheners, and I got lots of pictures and I spoke with her and everything. Right? So that's one way. If you're working with bacteria, that's another thing. Students like to work a lot with bacteria, but bacteria, the ones that cause disease in humans, they live around 37 degrees Celsius. So if you're going to work with bacteria, IB doesn't want you to work at room temperature. Not at body temperature, sorry. So you work with bacteria. And working with bacteria could be another area of concern. So it's about safety and about being humane. It's not about not being a good scientist. So if you want to use animals, just don't torture them. And you can even torture Daphnia if you put it in the wrong environment. Right. But you can still have beef for lunch or pork for lunch. They're okay with that. Which is an interesting discussion for you to raise, and IB students are supposed to be thinkers. So it's okay to challenge the IB and their policies, once you've got a good reason. And you've got, yes?
Why is it not, like, if eating animals is like cruel and stuff, like, how come plant eating is not?
And that's true. How come plant eating is, and how come there's no concern about plants? Do plants not have some kind of feelings? Are we discriminating? Is this because we belong to the animal kingdom? Are we hating on the plant kingdom? Right? So therefore, is it a cross-kingdom discrimination? You can ask that question if you want.
I can hear Sabrina's laughing.
Well, and also this is for Sabrina, who's not here today. So rock on, Sabrina.